Hello there. So, my name is Helen Geller, and I'm going to be your instructor this semester for Biology 1306. And if you are taking the lab as well, then Biology 1106 also. Here are my email addresses that you can reach me at, one for EPCC and one for UTEP, since I work there as well. So, just a reminder, if you haven't looked at the first video introducing you to the course, all pre-recorded PowerPoint lectures and supplementary materials such as videos are going to be found on Blackboard. Every chapter that we will cover also has a reflection assignment attached to it on Blackboard. And your exams will be found on Blackboard, as well as Pearson, MyLab, and Mastering, which is where you will find your homework assignments, which are multiple choice question assignments found on Pearson as well. If you are taking the lab as well, you will be able to find all of those assignments on Blackboard through Labster simulations. So if you're taking that lab, when you open up that course, I will have a separate video giving you instructions on how to access the Labster simulations. So we're going to go ahead and start with Chapter 1 from your biology textbook. In this chapter, it's really an overview of what we're going to cover this semester. So it's just going into the different aspects of what biology is. And so some of the themes we're going to be covering is looking at, what is life, what makes up the definition of when something is living. Some of the three unifying ideas of biology that we're going to cover would be looking at cells and how life starts as a cell and how life evolves, and then also the processes of information. So that's really looking at like DNA and RNA. So first things first, what does it mean to say that something is alive? Well, first of all living organisms share 5 fundamental characteristics. And those 5 fundamental characteristics are cells, where all living organisms are made up of membrane-bound cells. And we'll get into some more detail on cells later on. Replication, the fact that all organisms are capable of reproduction and replicating themselves, and they can actually do it on their own. This is important because when we talk about viruses cannot do this on their own, but living organisms can. And then finally, evolution. So looking at populations of organisms that are constantly evolving. So we're talking about, from the simplest cells of like bacteria all the way up to, what we are, humans. There are processes of evolution constantly happening. Last 2 things to define whether something is living is information. So really that's looking at DNA and RNA. It's hereditary information, your genetics, stuff that you're getting from your parents and your parents' parents and so forth. And then finally, energy. All living organisms acquire and must use energy. So thinking of, you have to eat, right? So you need to get energy from your food. And also this could be energy coming from sources that some organisms can do where they make their own food, like plants with photosynthesis. So I want to show you this video, and this one is showing you the first person to really kind of look at what cells were using his own creation, which is a microscope. So in this top picture, this is the original microscope created by Anton van Leeuwenhoek. And so it doesn't look anything like the microscope I'm sure you guys are used to seeing. But this is one of the first tools that was used to actually look at a cell on a microscopic level. And then the picture below is just showing you, what blood cells look like with one of our modern microscopes. So let's check out this video to explain the discovery of cells.
One of the great things about science is that when scientists make a discovery, it's not always in a prescribed manner, as in only in a laboratory under strict settings with white lab coats and all sorts of neat science gizmos that go beep. In reality, the events and people involved in some of the major scientific discoveries are as weird and varied as they get. My case in point, the weird history of the cell theory. There are 3 parts to the cell theory. 1. All organisms are composed of 1 or more cells. 2. The cell is the basic unit of structure and organization in organisms. And 3. All cells come from pre-existing cells. To be honest, this all sounds incredibly boring until you dig a little deeper into how the world of microscopic organisms and this theory came to be. It all started in the early 1600s in the Netherlands, where a spectacle maker named Zacharias Janssen is said to have come up with the first compound microscope, along with the first telescope. Both claims are often disputed, as apparently he wasn't the only bored guy with a ton of glass lenses to play with at the time. Despite this, the microscope soon became a hot item that every naturalist or scientist at the time wanted to play with, making it much like the iPad of its day. One such person was a fellow Dutchman by the name of Anton van Leeuwenhoek, who heard about these microscope doohickeys, and instead of going out and buying one, he decided to make his own. And it was a strange little contraption indeed, as it looked more like a tiny paddle the size of a sunglass lens. If he had stuck 2 together, it probably would have made a wicked set of sunglasses that you couldn't see much out of. Anywho, once Leeuwenhoek had his microscope ready, he went to town looking at anything and everything he could with them, including the gunk on his teeth. Yes, you heard right. He actually discovered bacteria by looking at dental scrapings, which, when you keep in mind that people didn't brush their teeth much, if at all, back then, he must have had a lovely bunch of bacteria to look at. When he wrote about his discovery, he didn't call them bacteria as we know them today, but he called them animalcules because they looked like little animals to him. While Leeuwenhoek was staring at his teeth gunk, he was also sending letters to a scientific colleague in England by the name of Robert Hooke. Hooke was a guy who really loved all aspects of science, so he dabbled in a little bit of everything, including physics, chemistry, and biology. Thus it is Hooke who we can thank for the term the cell, as he was looking at a piece of cork under his microscope, and the little chambers he saw reminded him of cells, or the rooms monks slept in their monasteries. Think college dorm rooms, but without the TVs, computers, and really annoying roommates. Hooke was something of an underappreciated scientist of his day, something he brought upon himself as he made the mistake of locking horns with one of the most famous scientists ever, Sir Isaac Newton. Remember when I said Hooke dabbled in many different fields? Well, after Newton published a groundbreaking book on how planets move due to gravity, Hooke made the claim that Newton had been inspired by Hooke's work in physics. Newton, to say the least, did not like that, which sparked a tense relationship between the two that lasted even after Hooke died, as quite a bit of Hooke's research, as well as his only portrait, was misplaced due to Newton. Much of it was rediscovered, thankfully, after Newton's time, but not his portrait, as sadly no one knows what Robert Hooke looked like. Fast forward to the 1800s, where 2 German scientists discovered something that today we might find rather obvious, but helped tie together what we now know as the cell theory. The first scientist was Matthias Schleiden, a botanist who liked to study plants under a microscope. From his years of studying different plant species, it finally dawned on him that every single plant he had looked at were all made of cells. At the same time, on the other end of Germany was Theodor Schwann, a scientist who not only studied slides of animal cells under the microscope and got a special type of nerve cell named after him, but also invented rebreathers for firefighters and had a kickin' pair of sideburns. After studying animal cells for a while, he too came to the conclusion that all animals were made of cells. Immediately, he reached out via snail mail, as Twitter had yet to be invented, to other scientists working in the same field, and it was Schleiden who got back to him, and the two started working on the beginnings of the cell theory. A bone of contention arose between them, as for the last part of the cell theory, that cells come from pre-existing cells, Schleiden didn't exactly subscribe to that thought, as he swore cells came from free cell formation, where they just kind of spontaneously crystallized into existence. That's when another scientist, named Rudolf Virchow, stepped in with research showing that cells did come from other cells. Research that was actually, how to put it, borrowed without permission from a Jewish scientist by the name of Robert Remak, which led to two more feuding scientists. Thus, from teeth gunk to torquing off Newton, crystallization to Schwann cells, the cell theory came to be an important part of biology today. Some things we know about science today may seem boring, but how we came to know them is incredibly fascinating. So if something bores you, dig deeper. It's probably got a really weird story behind it somewhere.
Okay, so a lot of scientists involved in this, and I think those are pretty good. These videos are pretty good at giving you an idea of everybody that was involved in these stories. And so some of those names you're going to hear a few more times throughout the semester as far as what they've contributed to science and specifically biology. So one of the terms that has been used so far is the word theory. And I know you've heard it before, but just to give you a specific definition of what it is, it's an explanation for some sort of a phenomenon, some sort of an observation that was made, but it has to be specifically supported by a wide body of evidence. There has to be a lot of support for it, for an idea to become a theory. So sometimes we just use theory, loosely in our everyday talk, but to use it specifically in biology, it has to be a very supported, highly educated guess or speculation as to what we think is happening. So some of the theories that you are going to be learning about this semester, first of all, cell theory. Okay, so this is one that in that video that was spoken about is what are organisms made up of. So we've got the cell theory. Then we have the theory of evolution by natural selection. Where do organisms come from and how are they evolving? And then the chromosome theory of inheritance. So how is hereditary information transmitted from one generation to the next? So how are we passing on our genes to our offspring and so forth? So we'll start off with the cell theory. So chapter 1 is really specifically just about cell theory, and we'll talk about cells later on in one of the later chapters as well. But to give you an idea, the idea or the concept of cells did not show up until the 1660s when you had Robert Hooke and Anton von Leeuwenhoek, just like the video had mentioned, were two of the most famous contributing scientists to look at a cell and come up with the concept of it. So by definition, a cell is a highly organized compartment. So it's a container, basically. It has a plasma membrane surrounding it, and it has stuff on the inside of it. Okay, so it's got a bunch of chemicals on the inside, and it is an aqueous solution. So basically, it's watery. And then we have the cell theory, which is stating that all organisms are made up of these cells, and all cells come from pre-existing cells. So that means, cells don't just pop up out of nowhere. Like in the video, one of the scientists had thought of cells came from spontaneously, they just happened. And that's not true. Based on cell theory, cells don't just pop out of nowhere. They're actually coming from other cells, from pre-existing cells. So in order to support the idea of cell theory and any theory, there has to be, experiments done. And so this is one of the most famous experiments to kind of, support the idea of cell theory. And this is by a scientist named Louis Pasteur. And he came up with a hypothesis after making an observation. And so a hypothesis is a testable statement that you can make after you make an observation. Then you would create an experiment where you actually test out that hypothesis that you have. A prediction, just kind of like, what do you think is going to happen? And so Louis Pasteur's hypothesis was that cells come from other cells, okay? And they're not spontaneously created. So this is what's leading into the idea of cell theory. So I'll show the video here in a second, but basically what you're seeing on the screen right now is a setup of Pasteur's experiment with two different types of flasks. So flasks is like a glass container that scientists can use where they could put a liquid in it or some sort of a substance, and they can make observations. On this particular straight neck flask, on the left here, Louis Pasteur had placed a nutrient broth inside of this flask. So a nutrient broth, think of like chicken broth or beef broth, like soup. It's got stuff in it. It's got food in it, basically. So he stuck it in a container, and he just let it sit there. And as he let it sit there, things that are floating around in the air would land in it because, as you can tell, the top of that flask is open to the environment. So particles that are floating around could go in there. So, it would be like cells, possibly bacteria cells or whatever else could be floating around, could land in there. So in the diagram, you'll see the little red dots would represent cells. Then he would boil it to kill anything that happened to be living in it. Just like, if you want to eat something, you probably want to boil it or cook it so that you kill anything that could be living off of it. And then after it would cool down, things that were floating around in the environment could re-land inside of it and start all over again. So his second experiment was to take a special kind of a flask called a swan neck flask. This one has that bent neck. So on the right-hand side, you see how the flasks have a very bent neck so that particles can't just land in it straight. So he placed a nutrient broth in it, which would have some cells just sitting in there already, boil it, kill all the cells, but now that neck with that bent shape, it's kind of an awkward shape, and it doesn't allow the cells, after the broth has cooled, to get back in there. They just end up settling in the neck and not actually getting down in the broth. So this seems pretty simple, but the idea here is that cells just don't pop up out of nowhere. They're coming from somewhere. So if you leave your food out, cells that are floating around in the air are going to land on that food, and then, bacteria, fungus, whatever's mold is going to grow. And so this kind of negated the idea of spontaneous generation, that life just comes from nowhere. So let's watch this video and they'll explain it better than me.
What can you do with sensitive skin?
Okay, what are they?
Cetaphil Gentle Skin Cleanser does more than clean.
In 1675, Antoine van Leeuwenhoek was actually given the honorary title of the Father of Microbiology. Anton van Leeuwenhoek was coined the Father of Microbiology because he observed the first motile meaning mobile or able to move around, so the first motile microscopic organisms. He called these organisms animalcules. He gathered these animalcules from a drop of pond water that he had collected. Van Leeuwenhoek also discovered the first bacteria. Cells had already been discovered by Hooke, but those were plant cells. Animal cells and bacteria cells were discovered by Van Leeuwenhoek. Before this time, microorganisms were hypothesized, but never actually observed. Many people were resistant to the idea that something could exist that was actually too small to be seen with the naked eye. It was not until the invention of the microscope that this micro world was able to be observed. Early microscopes did not have much magnifying power until 1675, when Anton van Leeuwenhoek developed the first microscopic lenses powerful enough to observe these microorganisms. These improvements resulted in a magnification of up to 300x. Louis Pasteur was known for creating a lot of different things, including our pasteurization processes that we still use today. Louis Pasteur performed a series of experiments that presented overwhelming evidence against the theory of spontaneous generation. Many consider Louis Pasteur's experiments to be the death blow to the theory of spontaneous generation. So first, some background. The controversy at the time was how microorganisms were able to grow in broth. Broth is a nutrient medium that has nutrients that microorganisms need to live. Broth had already been developed, and they had been using this to grow microorganisms in labs. It had been observed already that microbes were able to grow in nutrient-rich broth, but there was a debate as to why or how this occurred. Louis Pasteur postulated that microbes grew in broth because broth was exposed to microbes existing in the air, too small to be seen with the naked eye. These microbes would get into the broth from the air and then reproduce over a few days, growing enough to be seen. The other idea was in line with the theory of spontaneous generation. This idea was that the microbial growth observed in the broth after sitting out on a counter for a few days was due to something that was called the pneuma, or the vital force. The pneuma, or vital force, was like this spiritual force in the air that was able to cause spontaneous generation. So basically, these microorganisms were able to spontaneously generate out of the broth. In order to put these two different hypotheses to the test, Louis Pasteur created the swan-necked flask. The swan-necked flask was designed to allow air through, or the pneuma or vital heat through, but it would actually trap any microorganisms. Here we see a diagram of the experiment. At the beginning of the experiment, broth was boiled to make sure that there were no microorganisms existing in the broth at the beginning of the experiment. Then, the swan-necked flasks with broth were incubated for a few days. Pneuma, or vital heat, had access to the broth during this incubation period, but any microorganisms that would be in the air would not have access to the broth. After the incubation was done, they found no microbes, no microorganisms. The vital heat and pneuma was able to get through, but no microbes. So when they repeated the experiment, but this time they broke off the swan neck portion of this swan neck flask. These organisms that you couldn't see, that were just hypothesized to be in the air, now did have access to the broth. And they waited 3 days for the incubation, and lo and behold, there was growth in the broth. And so the microorganisms did grow, and the microorganisms were found at the end of the incubation. Pasteur's swan-neck experiments disproved the theory of spontaneous generation and gave overwhelming evidence that microorganisms exist.
Okay, so a little bit more of a background on cell theory, and so some of the review there about some of the other scientists involved, but specifically about Pasteur and his contribution to the cell theory. So what can we get from the idea of cell theory? Well, because we know that all cells come from pre-existing cells, we can also say that all individuals in a population must be related somehow by a common ancestor. And so all of the cells present in a multicellular organism means that they are also coming from pre-existing cells and must be related, through a common ancestor. And this then leads to the idea of evolution. And specifically, we're going to talk a little bit about chemical evolution. So I know you've heard of regular evolution, natural selection and evolution. But there's also the idea of chemical evolution, which is saying that there were specific chemicals available at the very dawn of the earth that allowed for life to come about. And so this chemical evolution had to lead to things being just perfect for life to come about. Okay, and so you have heard of Charles Darwin, I'm sure, and his idea of the theory of evolution. So in 1858, you had a couple of scientists, Darwin and another one, Alfred Russel Wallace, who's not quite as famous because Darwin ended up publishing a book faster than Russel Wallace did, stating that all species are related by a common ancestor. Okay, so like the idea of cells coming from other cells, you got to have that common ancestor. And then the idea of a species, which is a distinct, identifiable type of organism. So, like you have the human species, or like your dog at home is its own species, or a cat, or a fly, and so forth. Characteristics of a species can be modified from generation to generation, so little traits will change in the species over time. And Darwin called this idea the descent with modification. Okay, so changes over time. And here is another video about that.
Dishes still coming out dirty? Maybe your detergent. Only Cascade uses Dawn as a built-in pre-rinse system. It rehydrates dried-on food, lifts it off, and breaks it down, doing the pre-rinse for you. Cascade Platinum. Scrape, load, done.
This episode is brought to you by Squarespace. Everyone knows the name Charles Darwin. He was the father of evolution, the first to come up with that whole natural selection, survival of the fittest thing, right? Actually, Darwin doesn't deserve all the credit. While Darwin was puzzling over the beaks of Galapagos finches, another naturalist working in what's now Indonesia was reaching the exact same conclusions. His name was Alfred Russel Wallace. Darwin gathered the observations that led to the theory of natural selection while traveling the world on the HMS Beagle in the 1830s. After he returned to England, he spent decades developing his ideas and slowly working on a book about them. Wallace, who was born in 1823, was 14 years younger than Darwin. He was also less wealthy and less well-connected. While Darwin was methodically shaping the theories that would make him famous, Wallace was doing some globe-trotting of his own, studying natural history in South America and the Malay Archipelago. In 1858, he wrote a letter to Darwin, already a well-known scientist at the time, outlining in detail his ideas about how species changed through time. Needless to say, Darwin was pretty shocked to get a letter from some young nobody who'd come to the exact same conclusions about evolution that he had. Now, Darwin had been working on this book for a long time, and he did not want Wallace to publish first and get all the credit, so he and his friends quickly threw together a meeting of the Linnean Society of London, an association of biologists. In the meeting, the secretary of the society presented the ideas of both men at the same time. Wallace wasn't even at the meeting because he couldn't exactly commute from the remote islands in the Indian Ocean where he was working at the time. But neither was Darwin, because his 1.5-year-old son had died just a few days beforehand. Only around 30 people actually showed up at the Linnean Society to hear the papers be presented, and no one seemed to realize the significance of what they were hearing at the time. So, why is Darwin so famous today while Wallace is so obscure? Well, for one thing, Darwin had been working on his theory for decades. By a year after the Linnean Society meeting, he was ready to publish a pretty comprehensive book he'd written on evolution, on the origin of species. You might have heard of it. The book made a huge splash, and everyone pretty much forgot about the other weird young guy who no one had ever heard of. Remember, Darwin was already pretty famous. He'd written a wildly popular account of his travels called The Voyage of the Beagle over a decade before. It took Wallace another decade to publish his own book on natural selection, and by that time, the theory was already widely known as Darwinism. And when Wallace published another book on natural selection later on, he put the word Darwinism in the book's title. That probably didn't help. As the years passed, Darwin and Wallace had a polite but complicated relationship. Wallace believed that natural selection couldn't explain human intelligence on its own, arguing that some sort of higher power must be involved. Darwin and his friends didn't agree. But when Wallace ran into money troubles later in life, Darwin helped arrange for him to get a government pension in recognition for his scientific contributions. Today, Wallace is mostly just a historical footnote for non-scientists, an effort to raise money to build a bronze statue of him to commemorate the 100th anniversary of his death in 2013 only made it halfway to its goal, but poor Wallace hasn't been totally forgotten. He's considered to be the father of biogeography, the study of how species are distributed across the planet. His observations of how Asian and Australian species intermingled in Indonesia and New Guinea kickstarted scientific interest in how plants and animals end up living in specific places. The Wallace line, the surprisingly sharp boundary between islands with plants and animals more like those in Asia and plants and animals more like those in Australia, still bears his name. And in the end, they did make a statue of him, which was donated to London's Natural History Museum. And And maybe it's just as well that Darwin is the one who got permanently associated with the theory of natural selection, because Darwinism is just a lot easier to say than Wallaceism. But the contributions of Darwin's lesser-known frenemy Wallace still deserve to be remembered, not least because he maybe inspired Darwin to get going on actually finishing the book. Anyway, so you know that thing you want to do or are doing? Would it be better if it had a website? Well, this episode was made possible by Squarespace, which is a company that makes it pretty dang easy to create a custom.
Okay, so from that you got to hear about Darwin and Wallace, and we won't spend too much time talking about evolution this semester, but when you take 1307, there's a whole section that we get into evolution on. But this is just to give you an idea of, how theories work, and since evolution is important to biology, we want to make sure you understand some of the terminology. So we'll finish up our chapter today just going into a little bit more specifics on evolution. So again, evolution is going to be the change in characteristics of a population over time. And you need to realize that it is a population that is changing over time, not like an individual. Okay, so like let's say, I break my finger. That's not going to affect the human population because Helen, broke her finger. So it has to be a major characteristic that is happening amongst a population of organisms over time. And then that's going to be passed on, that trait. So what is a population? Well, it's a group of individuals of the same species living in the same area at the same time. So you could say the human population of the Earth, or you could say the population of humans living in El Paso, Texas, and that would be a smaller population because they're living in the same area at the same time. So the population of people living in El Paso might be different from a population, say, living in Norway. There's going to be some differences there in characteristics and health problems that you might be facing too. So that is very important to know that a population really has to be same species for sure, and then specifically in the same time and same area. So natural selection, as he had mentioned in the video, is really like the tool that kind of drives evolution. And two conditions have to be met in order for natural selection to actually occur in a population. First of all, individuals have to have some sort of different variations in a characteristic. And that characteristic has to be inheritable or passed on, be able to be passed on. So for example, hair color, eye color. We all have different eye color and hair color, but those traits there, eye color and hair color, are passed on, right? So you probably have your eye color and your hair color you may have inherited from your parents or your grandparents and so forth. Also, in a particular environment, that trait has to actually help the individual reproduce more versions. Okay, so hair color and eye color in a human doesn't really determine whether we're going to survive an environment, right? So it doesn't really matter. But hair color in an animal might be important for them to survive because, maybe it's camouflage. Maybe it's keeping them protected from predators. Or if they are a predator, maybe it kind of disguises them so that they can attack their prey. So that hair color might be very important for an organism that has to hunt and get food or be protected from getting hunted. And that trait might actually make them survive better in an environment. So if an inherited trait is going to increase your success in producing offspring, in other words, if that hair color on an animal protects it from getting eaten, then it can have babies, and those babies will have that same hair color, and they won't get eaten as often. So it's a good thing. And that trait will end up getting passed on through a population. So you'll start to see a population maybe only have that kind of a hair color that's going to allow them to survive. So in this way, the population's characteristic will change as a result of natural selection. So basically, nature selected that trait for that organism to survive with. And that natural selection actually acts on the individuals because, if an individual is born with a very dark fur and one individual is born with a very light fur, and let's say that they're living in a darker environment, the individual with the dark fur is going to survive and be able to pass it on. So nature selected for that individual to survive, and pass on its trait to its offspring. When an evolutionary change occurs in this population because of that trait, then you get a thing called speciation. And speciation means that when the populations are breaking off into a new species because of something like a trait that made it more beneficial, then you're just not going to see the other trait anymore, and it might end up turning into a new species. And we don't see that happening in larger animals right now, but in bacteria, there are always new speciation events happening because new species of bacteria are evolving off of other species. And then here's a couple of other terms that are related to natural selection. First of all, fitness. Like you've probably heard of survival of the fittest. Well, that's the idea of the organism that has the highest fitness, allowing it to survive in an environment, produce surviving offspring that can pass on those traits. So we're not talking like physical fitness, like, you got a good weight and you're nice and muscular and so forth. Fitness in this case is actually talking about the ability to produce offspring and that those offspring survive and produce other offspring. And then you have adaptation, which is the idea that a trait allows you to have better fitness to survive in a particular environment. So again, like that hair color, it might be an adaptation allowing the organism to have a higher fitness to survive in its environment because of its camouflage. So some examples of natural selection. Darwin had gone on this voyage to the Galapagos Island, and on those islands he saw finches, which are the little birds that we see here in El Paso flying around. So finches are just little birds, but the ones that he saw on the Galapagos Islands actually had different kinds of beaks. He noticed, they're all finches, right? They had the same kind of markings and the same size, but their beaks were very different. And he noticed that the food that was available on the Galapagos Island, there's different types. Some of them were like little soft, small seeds, and that was because there had been more rain. And so when he would see those soft, small seeds, he noticed that in that area, there would be finches that had very small, pointed beaks. And they did better because they were able to break open those small, soft seeds. And so he saw, these guys are actually doing really well. But if he had looked in other parts of the island, he might see finches with beaks that were much larger, and maybe they were eating something like fruit or larger nuts. And so the idea is that the beaks were very different on these different islands, and so it probably because they needed to eat different types of food. So going back here, the idea of cell theory and the theory of evolution by natural selection are saying when you put those two theories together, that all species are coming from pre-existing species, and therefore all species, past and present, must be able to trace their ancestry back to a single common ancestor. And again, you're going to hear more about this in Biology 1307, but that's just to give you an idea how these two theories relate to each other. Our last theory is the chromosome theory of inheritance. And this is going into, so we're talking about these inheritable traits and variations, right? And they get passed on. Well, how are they getting passed on? And that's going through chromosomes. It's going through our DNA and our RNA and being passed on to our offspring. So the idea of chromosomal theory of inheritance was actually proposed by a couple of scientists in the early 1900s, Walter Sutton and Theodore Boveri. Both of them came up with the idea of heredity and genetic information being encoded in something called genes. And genes, we know, are units located on chromosomes that contain information. And so it didn't happen until the 1950s when chromosomes were actually observed to the point where they can see that they were made up of another chemical called deoxyribonucleic acid, or DNA. And we now know that DNA is the hereditary material that's being passed on to offspring. And the genes then are actually chunks of DNA that code for certain cell products. So there might be a gene that codes for hair color inside the human genome. And that's a chunk of DNA that codes specifically for hair color. So we also have Watson and Crick, which are two other scientists. They came up with the idea of what DNA was and what it looked like and what it was made up of. So this is a picture showing you a model of what DNA would look like. And you've got these letters here. You've got some A's and T's and G's and C's. And you can see how the A's and the T's are hooked together. Sorry. And the C's and the G's are hooked together. And that's because they are actually bonded together because they are complementary to each other. And we'll talk more about DNA later, but just to give you an idea, we know that DNA is a double-stranded double helix made up of 4 building blocks, so those A's, those T's, C's, and G's, which actually code for some other chemicals called adenine, thionine, cytosine, and guanine. And the sequence of those letters is kind of like letters in a word, and it's a code, and it carries a code for information on what an organism needs to grow and reproduce. The 2 strands of the double helix are held together by connections. So those A's are paired with the T's and the C's are paired with the G. And this pairing allows DNA to get copied later on and passed on to offspring. So here you go, you see the A's and the T's and the G's and the C's. Okay, and so a couple other things here. The Tree of Life is also saying that we've got 3 major groups of organisms. We've got our eukaryotes and our prokaryotes. The eukaryotes are what we are, humans, all the animals and plants. We all fall under the eukarya group of organisms. And then the other 2 groups of organisms are called prokaryotes, and that's going to be all your bacteria. And so there's 2 groups of bacteria called bacteria and archaea. And we'll talk more again about all of these, but bacteria are going to be like the bacteria that we run into on a daily basis, that's growing on our skin and growing on the food that we don't take care of and so forth. And the archaea are going to be the bacteria that we don't really have much connection to physically on a daily basis, but they live in really extreme environments, say like in the deep ocean vents of the deep vents in the ocean that are spewing out hot water, or in volcanoes, or in the Dead Sea, the Salton Sea. They live in very extreme environments, but both of them, archaea and bacteria, belong to this category called prokaryotes. And so I'm going to show you another video here showing you what eukaryotic cells and prokaryotic cells look like and how they're related and how they're different.
On Earth, there are 2 major types of cells, prokaryotic and eukaryotic. Prokaryotic cells are the simplest and most ancient types of cells. They were the only form of life on Earth for billions of years before eventually giving rise to eukaryotic cells. If you take a look at prokaryotic and eukaryotic cells side by side, you can see a number of differences. To begin with, prokaryotic cells are a lot smaller than eukaryotic cells and have a simpler structure. But bear in mind that this simple structure is actually a good thing for prokaryotes because it allows them to reproduce very quickly and very effectively. If you were to look inside a prokaryotic cell, you would probably be surprised at how simple it is. For example, prokaryotic cells lack a nucleus. In fact, the name prokaryote actually gives you a hint about its structure. Kary means kernel or nucleus, and pro means before. So basically the word prokaryote means before nucleus. The inside of the prokaryotic cell is basically an open unit with no compartments. There are no membrane-bound organelles. However, small structures called ribosomes are scattered throughout its cytoplasm. The cell's DNA is located in a region of the cytoplasm called the nucleoid region. The nucleoid region is not the same thing as a nucleus because it's not enclosed by a membrane. Prokaryotes also have a cell wall that surrounds the plasma membrane. The composition of the cell wall depends on whether the cell is an archaean or a bacteria, but basically it consists of complex polysaccharides. Prokaryotes also often possess 1 or more flagella, which are used for movement. In contrast, eukaryotic cells contain a membrane-bound nucleus and other membrane-bound organelles. The name eukaryote also gives a hint about its structure. Eu means true, and remember that kary means nucleus, so in essence the word eukaryote means true nucleus. Eukaryotic cells are larger and much more complicated than prokaryotic cells. However, they do share a few things in common with prokaryotes. Let's take a look at a Venn diagram to compare prokaryotes to eukaryotes and to also see what areas they have in common. Let's start with their commonalities. Both prokaryotes and eukaryotes have DNA as their genetic material, have ribosomes, have cytoplasm, and have a plasma membrane. Now let's look at some of the ways they differ. Prokaryotic cells are the oldest type of cell. They're small and relatively simple. Eukaryotic cells evolved from prokaryotic cells later. They're larger and much more complex internally. Prokaryotes lack both a nucleus and membrane-bound organelles. Eukaryotic cells have a nucleus and also contain organelles. Prokaryotes are single-celled organisms. Eukaryotes can be either single-celled or multicellular. And finally, the DNA of a prokaryote is usually organized as a single circular chromosome, while the DNA of a eukaryote is organized as linear chromosomes.
Okay, so you can see the two cells. We're going to spend more time talking about cells in a couple of chapters, but you can see a lot of differences between the two different types of cells. And again, eukaryotic cells are going to be the ones that we belong to, so animals and plants. And then the prokaryotic cells are going to be the ones that are like your bacteria. Okay, so the next part here is taxonomy. Taxonomy is really a system that's been designed to help make biology a lot easier because it gives everything a name. So it's designed to give all organisms, big and small, its own unique name, and also classify those organisms into their own groups. So a taxon would be a named group, hence the name taxonomy. When you have a taxon, it kind of makes things easier for any person who's looking at an organism to identify, oh, this is a mammal, or this is a bird, or this is, a fish or whatnot. So taxons are great. Taxonomy is a very useful system. When you're looking at taxons, one of the main taxon or group is called a domain. And there was a scientist named Woese who created this taxonomic level domain, and there's actually 3 domains, which are bacteria, archaea, and eukarya. So those should sound very familiar because we just spoke about the different types of cells being either bacteria, archaea, or eukarya. So bacteria and archaea are going to be your prokaryotic cells. So that's all your bacteria. And then eukarya is going to be all the eukaryotic cells. So that's, again, what we as animals belong to. A phylum is going to be another category underneath domain. Okay. So where did this idea of taxonomy come from? Well, in 1735, so a long time ago, before all this stuff with DNA and everything, we actually had a scientist named Carolus Linnaeus who established a classification system that he started this, 400 years ago now, or almost 400 years ago now, and we are using it still today. So each organism in this system actually gets 2 names for a scientific classification. So that consists of the genus and the species. A genus is going to be all your species that are closely related to each other. Okay, and then a species is going to be a unique individual that can only breed with another individual of the same species. So the species are going to be the individuals that have the same characteristics, and they're very distinct from other species. So for example, your dog at home is a species, and then a coyote or a wolf is another species, okay? Or cat, your house cat is one species, and a lion would be another species. So there are certain rules of naming your organisms. So this is called nomenclature, when you give a name to an organism. So the naming system consists of the genus and the species. And we call this a scientific name or a Latin name because most likely the scientific names are going to be in Latin. always, the rule of thumb is scientific names are always italicized. Back before computers and really good typewriters, they used to just underline. So italicizing, if they couldn't do it with a computer, even nowadays you would just underline the name. Genus names are always capitalized, and the species name is never capitalized. So for example, Homo sapiens, which is our species, Homo is the genus, it's capitalized. Sapiens is a species, not capitalized. And look, they're both italicized. Okay, so we're going to wrap up this chapter with the idea of doing science, okay, and what it entails. So we also could call this like the scientific method, which I'm sure you guys are familiar with. So we know that scientists ask questions, and they want to answer those questions by collecting data. And so science is about formulating a hypothesis or a guess, and then finding evidence that supports or conflicts with that hypothesis. So for example, you can use designed experiments, like some of the ones we mentioned in this chapter today. Biologists will test ideas about something that they witness, and then you make the predictions and you test those predictions, and you can actually have an alternative hypothesis that may be true if things aren't working out the way that you had hypothesized them to be. So hypothesis testing is a two-step process. First, you state the hypothesis as precisely as possible, and you make a list of predictions that it can make. And then you design an observational or an experimental study that can be there to test those predictions. So you want to make sure it's a very clear hypothesis, and then you want to make sure that your experiment would be able to test it to see whether it's supported or not. So one of the famous hypotheses from the past is about the idea of why giraffes have long necks. So there was one hypothesis presented by Simmons and Scheepers that said that the reason why giraffes have long necks is because they had evolved them so that they could reach food that was very high up in trees that would be unavailable to other mammals. So their prediction would be neck length is variable among giraffes, so that means that it's different among different giraffes. Neck lengths in giraffes is heritable, which means it's passed down, and that giraffes feed high up in trees, therefore supporting the idea, they have to have a long neck to reach the food at the top of the trees. So Simmons and Scheepers tested this idea out, and they actually found out that that third prediction, that giraffes feed high in trees, was not true. They actually found out that the giraffes don't feed high up in the trees. They'll feed from any part of the tree that they want to. So that kind of threw out that idea. So their alternative hypothesis would have been better to support that. So here's a little graph, and it kind of shows you males and females and their average height, and also where they're feeding at in the tree. And you can see, even if they had to feed high in the tree, their neck is actually way too long to even need to worry about having a high tree, because the trees where they're at are not that tall anyway. And so they could feed in the middle of the tree, they could feed at the top of the tree, but really it was not about the height of where they were feeding from that made why their necks are long. So the alternative hypothesis was actually that giraffes had longer necks, especially specifically the males, because they would fight with other males to compete for the females. So it was actually a reproductive competition. So the longer necked males would actually fight. And if you've ever seen this, and you can look it up, male giraffes will fight with each other with their necks. It's like they're sword fighting, but with their necks, and they're like whacking each other. And the ones that have the longer necks would win. And therefore, if they win, the female will choose to reproduce with them, and then have offspring, and their offspring would have that trait of longer necks. So there's plenty of data to support this hypothesis now, and therefore it refutes or says that the food competition hypothesis is wrong. But still, this is one of those kind of things that could still be tested. I mean, there could be some scientists right now testing this theory, or this idea rather, and they may say, you know what, it's not for competition for reproduction, it's actually this. So hypotheses are always tested over and over again. Things are always challenged. So that's something that we have to keep in mind. Things get changed a lot. So the whole idea of experimental design is, you make these decisions based on high selection of evidence. You're asking questions, you're posing hypotheses, and you're doing all of this to try to see which hypothesis is supported or not. So that's where we're going to end this chapter 1. Chapter 2, we will cover more about chemistry and biochemistry. So thank you for listening. Hope you guys enjoyed this, and just be sure to rewind the video if you need to and go back, and you could look at the clips of the videos that were embedded in here as well on your own. And thank you again.